video we are going to learn about cyclic photophosphorylation. Now in the previous video we have learned about non-cyclic pathway or non-cyclic photophosphorylation. Here we are going to discuss the cyclic pathway or cyclic photophosphorylation. So before that, let's just recap what did we learn in photosynthesis. So photosynthesis. Photosynthesis is completed in two reactions. The first one is called as light reaction, and the second one is called as dark reaction. Now the light reaction is also known as light dependent reaction because it is dependent on light, and dark reaction is called as light independent reaction because it doesn't need light, but if there is light also it can do its function. If there is no light also it can do its function. Now light reaction is completed in two different pathways. The first one is called as non-cyclic photophosphorylation and the second one is called as cyclic photophosphorylation. So this thing we have discussed in the previous video, non-cyclic photophosphorylation or non-cyclic pathway. So this light reaction is also known as photophosphorylation. So the photophosphorylation or light reaction is divided into two types, non-cyclic and cyclic. Non-cyclic is where ATP and NADPH molecules are synthesized in the presence of light, and the electrons which are excited, they never come back to its own to the photosystem where it has started. So that is not the case in case of cyclic. It comes back again. So we are going to discuss this cyclic photophosphorylation in detail in this video. This dark reaction is called as C3 cycle or Calvin cycle. So here we are going to focus on cyclic photophosphorylation. So cyclic photophosphorylation now, when we have looked at non-cyclic photophosphorylation or non-cyclic pathway in the previous video, we have learned that non-cyclic pathway starts with PS2 or photosystem 2. In case of cyclic photophosphorylation, it basically starts with PS1. So that is the only difference. So this is our chlorophyll A molecule, or maybe let me just draw it with red. So this is my chlorophyll A. This is the chlorophyll A molecule which is present in the middle, and here are the accessory pigments. Like this. So this is my chlorophyll A molecule, and this is called as reaction center chlorophyll. So this is called as reaction center chlorophyll. Because why is it called reaction center chlorophyll? Because here is where the main reaction of photosynthesis happens. What about these? These are called as, so these pigments other than chlorophyll A, apart from chlorophyll A, all other pigments are present around it or scattered around it. They are called, these pigments are called as accessory pigments. Accessory pigments. So this is our photosystem 2. So here if you label the main thing, photosystem 1, so this starts with PS1, and we know that PS1 absorbs 700 nanometer. So 700 nanometer, PS1 or photosystem 1 absorbs 700 nanometer. This is the most important thing. That is the only difference, because in non-cyclic photophosphorylation, the whole process starts with PS2, which is photosystem 2. Here in cyclic pathway, it starts with PS1, which absorbs 700 nanometer of wavelengths. So what happens now? Here we have the sunlight. This is the sun, so S, and from the sun there are these wavelengths which are falling on the photosystems. The light energy is falling on the photosystems like that. So the light energy comes in the form of particles called photons. That this also we have looked in the previous videos. So photons are basically the light particles which travel from sun and fall on this photosystem. So photons come and fall. Then what happens is the photons fall on the outer surface. So they fall on the outer pigments which are present at the outer side. So for example, it fell on this pigment here. So this pigment is going to transfer the energy or pass on the energy to the inner one, like that. So this thing also we have looked at when we are talking about quantosome and photosystems. So again, here as well, the pigment molecules which are present on the outer surface, they pass on the energy from the sun which they are getting. They pass on the energy to the inner particles, pigment particles. So finally, all the energy from the sun goes to chlorophyll A, and that's why it's called as reaction center chlorophyll, because here is where the main reaction occurs. Now from here, when the photons fall on this, when the light particles or energy particles from the sun fall on the chlorophyll A, from chlorophyll A, because from chlorophyll A has got all the energy from the sun. So finally all the accessory pigments have passed the energy to the chlorophyll A. Now when this chlorophyll A has got all the energy, a pair of electrons, so let me write this on the other side. These are photons. So a pair of electrons from chlorophyll A is going to get excited and move upwards. So it's going to get excited upwards. So this is my pair of electron which gets excited upwards, and it goes to something which is called as PEA, or primary electron acceptor. So here you are going to write the abbreviations. So PEA is primary electron acceptor. So a pair of electrons is going to get excited from this chlorophyll A molecule, and it goes to primary electron acceptor, where this electron acceptor is going to accept the electrons. It's going to act as like a receptor, and it's going to accept or take the electrons and pass on to the next electron acceptors which are going to come here. So from primary electron acceptor, the electron goes here to primary electron acceptor, and from here there is one more electron chain which is going to come from this main primary electron acceptor, which has Fd as its electron acceptor. What is Fd again? Fd is ferredoxin. Ferredoxin is also a small little electron acceptor which is present here. Now from this ferredoxin, so again if we put the electrons here, because from primary electron acceptor the electron is traveling, the pair of electron is traveling. So from this ferredoxin electron acceptor, it's going to go to plastoquinone, and from PQ it is going to go to CY, and we know that CY is cytochrome. Again, from cytochrome it is going to go to finally to PC, and PC is plastocyanin. So the pair of electrons from primary electron acceptor is going to travel to ferredoxin, and from here it's going to go to PQ, which is plastoquinone. So this is a pair of electron, and from PQ also to cytochrome, from plastoquinone to cytochrome, and from cytochrome to plastocyanin. Also the pair of electron is going to travel there. Now when it is going through FD, which is ferredoxin, when it is going through FD, the extra energy which is there is used to combine one ADP molecule with an inorganic phosphate molecule to form ATP. What is ADP? ADP is adenosine diphosphate and P is inorganic phosphate molecule, and they are both combined to form ATP, which is adenosine triphosphate. And this is a primary carrier for energy through living systems. So ATP is the energy currency of the cell, and we use ATP, or the plants, they use ATP to produce energy. So ADP is combined with an inorganic phosphate molecule to form ATP, and this is what the main thing that we require in light reaction. So in non-cyclic photophosphorylation, which we have looked in the previous video, there ATP is synthesized and NADPH is synthesized. Here just ATP is synthesized, there is no NADPH which is going to synthesize. So here when it is going through FD, ATP is synthesized, and when it is going through CY, which is cytochrome, also one ADP molecule is going to combine with an inorganic phosphate molecule to form ATP. So two times ATP is getting synthesized here. One when it is going through ferredoxin, two when it is going through cytochrome. The extra energy is used to combine these both ADP and P, which is inorganic phosphate molecule. So here at ferredoxin and cytochrome, ATP both two times. So ATP is synthesized two times here in cyclic photophosphorylation. So some important things that we need to remember. So I'm going to erase this thing here. So from plastocyanin, so finally the electrons are reached to plastocyanin. So it went again. Let's recap. So from chlorophyll A, it went to primary electron acceptor. Why is it called primary electron acceptor? Why primary? Because it is the first electron acceptor which is going to accept the electrons, and then after this ferredoxin came. So this is not primary. Maybe it could be secondary, because this is not the first one to accept the electrons. The first acceptor to accept the electrons is this one, primary electron acceptor. Next, FD, ferredoxin. From ferredoxin, here ATP synthesized, and from ferredoxin it is going to go to plastoquinone, and from plastoquinone to cytochrome, where ATP is getting synthesized, and from cytochrome it's going to go to plastocyanin. Now from this plastocyanin, here now where is the pair of electron? The pair of electron is near plastocyanin. Now that pair of electron is going to come back to this photosystem, and so here the pair of electron is going to come back to this photosystem, and that's why we call this cyclic photophosphorylation. Why is it called cyclic? Because the whole process of photosynthesis has happened from here. So the electron started from sun. So sun, because we are taking only the chlorophyll or photosystem into consideration, so the photons came and they fell here on the chlorophyll A, and chlorophyll A has the energy, extra energy. So in the atoms of this chlorophyll A molecule, the electrons in those atoms get excited and they go to FD, PQ, ferredoxin, plastoquinone, cytochrome, plastocyanin, and from here the pair of electron goes to come back here again, because it started from chlorophyll A of PS1, which absorbs 700 nanometer. Again it is coming back to here. When we looked at non-cyclic photophosphorylation, there the pair of electron never came back to its to the position where it has started. So here it's not like that, it's another case. So what happened here, it started with chlorophyll A of PS1, which absorbs 700 nanometer. Again it came here and it came back to its own position again. That's why it's called a cyclic photophosphorylation. Why cyclic? Because it's like it's in the form of a cycle. So cycle, again it's coming back here like that. Now the important things that we need to remember about cyclic photophosphorylation are, the first point is only PS1 is involved. Only PS1 is only involved. There is no PS2, because why do we need why we don't need PS2? Because the pair of electron is coming back to PS1, so there is no need of PS2. So PS1 is only involved. There is no involvement of PS2 in case of cyclic photophosphorylation. The next thing, there is no photolysis of water. Now if you have remembered about photolysis, we have learned that in the previous video. Photolysis is the splitting of water. So photolysis, so let me write that equation again here for you. H2O, which is water, gets splitted into a pair of electron and a pair of proton and an oxygen molecule. This pair of electron goes back to here, and oxygen molecule is as a byproduct, and the pair of protons, they go to NADPH in non-cyclic photophosphorylation. Here there is no such reaction which is taking place. There is no photolysis of water. Why there is no photolysis? Because here, if you notice carefully, the pair of electron has started its journey here, and the pair of electron afterwards, after finishing its journey, it's coming back to its same place. So if you think this as a home, this as the electron's home, it's coming back to its home at the end of the day. So because that's why we're calling it cyclic, right? So that's why we don't need photolysis of water. Actually, what is the reason? Why in non-cyclic photophosphorylation there is photolysis of water, or why there is splitting of water? Because there is a constant flow of sunlight on the photosystem or chlorophyll A molecule, and constantly the electrons are getting lost. So there has to be some mechanism to replace the electrons, right? So that's why we use this photolysis. Here there is no such kind of thing. There is no such kind of photolysis or splitting of water which is taking place, because the electrons are coming back to its own position where they have started its journey. That's why there is no photolysis, because the main purpose of photolysis is to replace the electrons. Here no need to replace electrons, because it has those electrons. Why to replace it? So that's why there is no photolysis of water which is taking place. The third important point is only ATP is synthesized, or only ATP is made. So synthesize. So usually in light reaction there has to be two products which has to get synthesized, ATP and NADPH2. Here just ATP synthesized, when it is going to ferredoxin and when it is going through cytochrome. In the previous video we have learnt about non-cyclic pathway. There ATP synthesized and NADPH2 is also synthesized. Here there is no NADPH2 which is getting synthesized. That's why one more important thing, there is no reducing power. No reducing power. Now what do I mean by no reducing power? When we have looked at non-cyclic pathway, there when it went here and when it went to PS1, there this had the at PS1 it went to the primary electron acceptor, and from primary electron acceptor it went to ferredoxin electron acceptor, and from ferredoxin NADPH, and there it gets reduced. Here there is no NADPH which is forming. That's why there is no reducing power at all. So that NADPH and the proton, the pair of proton which we have got here, that pair of proton goes to that NADPH to form NADPH2, because that's what we need, NADPH2 and ATP. Those are the two things that we need after the light reaction. So that's why there is no reducing power or no NADPH2 which is being formed. So 4 points, PS1 is only involved, there is no PS2 involvement, no photolysis of water, and only ATP is synthesized and no reducing power. Now the next thing is the location or the site where this cyclic photophosphorylation is going to take place. So cyclic photophosphorylation takes place in the stroma lamella. Now, cyclic pathway takes place in the stroma lamella. What is stroma lamella again? So, I'm going to put here. So, if this is the chloroplast organelles in a plant cell. So, these chloroplasts are double membraned and they have thylakoids, which are the coin or disc shaped structures which are present in the stroma, which is the fluid matrix, and the thylakoids are stacked onto each other, and that stack is called as granum, or plural is grana, and these two grana are attached to each other by some interconnections like this. This is nothing but stroma lamella. So this thing here. So either they are attached to two grana, or either they are attached to one granum and to the membrane of the chloroplast like this. So they are attached to the membrane like that, or between two grana as well. They are just interconnections which are there to hold those grana in place, like in a stroma. So we'll draw one more. So this is nothing but stroma lamella that we are referring to. So this is nothing but stroma lamella, those interconnection thread-like structures. So this cyclic pathway, this cyclic photophosphorylation takes place in this, on the membranes of these stroma lamella. So here is where it's going to take place. So here we have learnt about cyclic photophosphorylation. So why is it called cyclic? Because the electrons are coming back to its own position where they have started, not position or the photosystem. In non-cyclic, it started with PS2. The pair of electron never came back to PS2 again. So it went through PEA, which is primary electron acceptor. It went through plastoquinone, cytochrome, plastocyanin, and it went to photosystem 1. From photosystem 1 it went to primary electron acceptor, and from there ferredoxin, and from there the NADPH is getting reduced. That's why we told there is reducing power in non-cyclic pathway, and from there a pair of proton went and there it gets reduced and forms NADPH2. So this is about cyclic photophosphorylation or cyclic pathway, and we have looked at some of the important points. PS1 is only involved, there is no photolysis of H2O or water, and only ATP synthesized, there is no NADPH2 and no reducing power because there is no NADPH2. And the fifth point is the site where this cyclic photophosphorylation takes place. It takes place in the stroma lamina, this one here. The interconnections between two grana or one grana to the membrane of the chloroplast. So this is all about cyclic photophosphorylation.